All right, let's get started and welcome back. It's been a while, and I will just first, as I said, briefly, I'll try to be brief, talk about the experimental design because I assume that everyone watched it. So I will only briefly talk about some of the important parts, okay? So doing experiments, we have this kind of research circle. And what we are trying to do here is to say, well, if I have a experimental idea and if I have a hypothesis that I want to test, how can I test it? How can I test it experimentally? So this is why we need experimental design. So here I just skip, I would only talk about the important parts. So from idea to hypothesis, this is the most important part, right? Because why on earth we design experiments? Because we want to answer research questions. And what are the research questions? What are the research ideas? Where do they come from, right? Because sometimes, most of the time, you worry or you wonder about that. You see a paper, hey, this is so cool, but where did this idea come from? So there can be some existing theory. So if you read a lot of previous literature, there are papers from 10 years ago, 20 years ago, 50 years ago, and then they talk about the same thing. And there are some research gap that is unresolved, right? There's literature, and then a lot of people, they work on the same topic, but some tiny bit of the knowledge is not known yet. So this is the rich research gap, and then you want to do it, and you want to solve it. And another one is from also personal experience. So regarding to decision making, and this is quite relevant to our everyday life. And you could imagine that, well, if you are facing this chocolate brownie and the smoothie decision problem that we had the last time, and you might wonder, every time if I encounter this problem, I maybe have a difficult time in making my decision. How does that work? So I'm interested in decision making. I want to design experiment to test how decision making works in this case, right? And when we have the research idea and when we have the hypothesis, and well, what can be a hypothesis? Hypothesis is something that you could anticipate or you theorize the reason why we make the decision, for example. So in a hypothetical example, again, the chocolate brownie and the green cucumber smoothie or broccoli smoothie example, you might wonder that, well, this is the decision problem. People have their own preference. Some people, they may prefer sugar. Some people, they may prefer a healthy lifestyle. So the hypothesis can be lifestyle might affect how people make food choices, right? This sounds already quite scientific. And this is the hypothesis. And what do you want to do with the experiment, right? You have a kind of a list of questions to ask the participants. So what do you do for life, to ask the lifestyle. So when do you get up? When do you eat breakfast, lunch? Do you eat breakfast? Do you eat lunch, dinner? And when do you eat it? Do you have a regular schedule? And do you do a lot of sports, or do you do whatever? This kind of questions, right? And then you have a summary of the score. And then this is the summary of the lifestyle of the person. And when you have the questionnaire of the lifestyle of the person, you then ask the participants to make repeated choices. And then the choice can be chocolate brownie versus the smoothie. It can be something like pizza versus, I don't know, yeah, whatever, some healthy things. Avocado, for example, repeated choices. And then you could make a simple correlation, right, to see if the healthy lifestyle or the lifestyle in general, healthy versus unhealthy, could affect food choice in terms of the calorie and the protein and also stuff, fat, for instance, sugar, right? This is quite an interesting experiment already. So I guess this is the one quite hypothetical example that I just come up with on the fly. I didn't think about this when I prepared for the lecture, but I think this is something that you can really understand. Okay, so then back to here. Experiments why we do experiment, because we want to establish some causal link, and we also want to control many of the variables. So here I give you the example of chocolate brownie versus smoothie, and then this is another example. The hypothesis is how the positive mood of people will affect their intellectual performance. This is hypothesis. How do you want to test it? Same idea. You get the mood and then you measure their performance. Similarly, as what we had before, earlier, I said people's lifestyle and their healthy food choice, right? This is the link. And what you can do here in this particular case is that you have 40 healthy participants that come into the lab, and then they didn't know what they will receive. So here this randomization. So half of the people, they will be assigned to the funny, positive mood group, and the other half will be assigned to the control group, the ordinary. So the idea is assuming that when they come, before they come into the lab, there is no difference. And then we do randomization to randomly assign people to these two groups of treatment. And then we measure their performance to see if there is a connection. So what we want to know is what are the variables, what are independent variables, and what are dependent variables. So independent variable just means what we manipulate, and the dependent variable is what we want to measure. So in this case, the independent measure is the group, the treatment assignment, positive mood versus neutral mood. And then the dependent variable is the kind of a questionnaire or a test that they will have a score on. So then here I will skip, and randomization, it is very important. So why we do randomization? It is because there might be some other factors that might affect the behavior of the participants. So people's time of the day when they come in to do the experiment is different, temperature is different, the noise is different, and a lot of things are different, right? And what we want to do is we want to balance it so that if we randomize that pretty much, if we randomize the group assignment enough, so all of these nuance or undesired variables, they will be balanced out. So this will be a way to control for the confounding variables. So randomization is very important. So if, let's say, we do not do a randomization in this case, randomization, so let's go to one step back. So randomization, what can be done is there's a person coming to the lab, I have a coin. If it is the head, and then the person will go to the treatment group, positive mood. If that is the tail, if the coin is the tail, then the person will go to the control group, right? Randomization. But what if the first 20 people they come in, they will both, all of them will go to the positive mood group, and then the second group of 20 people, they will go to the control group. This is quite biased already. So this is not a good randomization. Because you as an experimenter, you might have a bias towards the participants when they come in. So this exercise we could skip, and then type of experiment I will also skip, and then this is important, I will talk about it a little bit. So there are a few important types of experimental design. So one idea is if, for example, I want to test a way of education, how that could affect the performance of students. So I will, for example, on psychological statistics, for example. So if I have a new way to teach statistics as opposed to the more traditional way. So there are two teaching methods. Let's say one way is the new teaching, the other one is the old teaching. So, and how can you measure the change, right? How can you measure the change? So presumably you will pick two classes, and one class they will receive the new teaching method, and then another class, they will receive the old teaching method. But you want to also make sure that at the beginning, there's a baseline. So you will at the beginning of the experiment, before you implement your teaching method, you just go to the two classes, give them kind of an exam, and then ask them to take the answers at the beginning of the semester. And then this score at the very beginning of the semester will be the baseline. During the semester, there will be the teaching, there will be the implementation, and then the teaching will just teach old methods versus new methods. And then at the end of the semester, and you will have to take the exam again, maybe different questions to test if the students understand the concepts and also some knowledge they have gained through different teaching methods. And what is the key here? So the key here is there is the within subject parts. So within subject means repeated measure. So one person will get a measurement more than one time. So if I am in the new teaching classroom, I will be tested at the beginning of the semester. Me, as the same person, I will be tested a second time at the end of the semester. So this is a within subject, within myself, I am tested twice. And then there's also the between subject part, meaning that I am a person from classroom A, and then there is another person from another class, and then I receive the new method, and then they receive the old method. We cannot receive old and new method all at the same time, so it's only one person, one class for the new method, and then the other class they receive the old method. So this is the between subject parts. So each group they receive different treatment or different manipulation. Okay, so here I have a skip, I guess you get the idea, between and within, and this also can be mixed together. So the one I told you is already a mixed design. So there is the between subject factor, class A, class B, and then there's also the within subject factor before and after the teaching. So at the end and also at the beginning of the semester. This is a mixed factorial design, and that there are two factors, right? Beginning of the semester, end of the semester, and here teaching method, new teaching method, old. So this will be then a 2x2 design. If you read a lot of papers from the literature, you will see how they describe their experiment. 2x2, 2x2 is the most common one, honestly. And 2x3 is also common. And sometimes there are more complicated experimental design, like 3 levels. 2x2x2, for instance. So in total, it's 8, right? There will be 8 sub treatments. Yeah, so then here I will skip many of the things. And at the end, I think it's analysis. Only 1 minute. So it depends on what you want to measure. There will be t-test, ANOVA, and the correlation. And at the end of this lecture that you watched, I also talked about the importance of computational modeling. This is maybe a little bit new to you, but this is very helpful and powerful in terms of how we could use models like formal mathematical formulations to try to understand human behavior. Why am I talking about mathematical modeling? It is because mathematical modeling or computational modeling, not only because I use it, it's because this is closely linked to decision neuroscience. So if you look at the decision neuroscience literature, I would say more than 60 or even 70% of the paper, there is some kind of computational modeling. So this is important that you are aware of it. All right. This is my supposed to be short, but not so short recap of the experimental design session. Any questions? When I talked about it or also when you were watching it? No? Good? Okay, then I guess assume you don't have question. So let's then move to the next. Wait a sec. All right, so if you are all familiar of experimental designs, the next question is, what are the measurements most of the cases? So if we have decision neuroscience, we could design experiments to answer research questions regarding decision making. But where is the neuroscience part? What can we do to measure? How can we measure the brain? Are there different methods? And what are the advantages and the disadvantages regarding each method? So we will now talk about the research methods in decision neuroscience, but also this also applies to cognitive neuroscience in general. So if you study vision, if you study language, all of the method, I think they will apply. So in essence, we first want to answer the question, what is decision neuroscience? And there are two words, it's simple. One is decision, the other one is neuroscience. So what you could imagine is decision neuroscience simply means the neuroscience or the neuroscientific research of decision making, right? So two parts, decision making and neuroscience. And from a few years ago, and then there is a kind of emerging term that happens in the literature or in the field. It is called neuroeconomics. And neuroeconomics is actually more or less equivalent to decision neuroscience. Economics is a larger field. They study everything, but neuroeconomics is the smaller one, especially it studies it uses neuroscientific approaches to study microeconomics. And then this is more related to human behavior, maybe rational, maybe irrational, purchase, economic choices. And then some of economists, they approached neuroscientists, and they wanted a collaboration. And then this is how everything gets started. So if you ever encounter the term of neuroeconomics, it's 90% of the time means decision neuroscience, the same thing. And why we have this field, why we have decision neuroscience, why we have neuroeconomics, because there is a goal that we want to achieve. So this is the central research question that we are interested in. So here the research question is to understand the process that connects sensation and action by revealing the neurobiological mechanisms by which decisions are made. So it's the same thing that I said, using neuroscientific methods to uncover the process underlying decision making, both in terms of behavior and also in terms of the brain. And neuroscience is a larger field. If you study the bachelor of neuroscience, you will spend 3 years learning neuroscience, and we can't cover everything in our lecture, but only just a small piece of introduction. Neuroscience in a nutshell. So what we do, we study the brain, right? And then there are multiple levels of the brain. So we have the entire brain as the organ, and then we have brain areas that we will encounter throughout the semester. And then what's next? So we have neurons, and then this is the cell of the brain. So we have, this is the unit of the brain. And what we also have, we have also like this, right? Neurons and the dendrite, for example. And we also have gray matter and red matter, and we also have different parts of the brain and midbrain, cortex of the brain, and the stem of the brain, and spinal cord even, that's also part of the central nervous system. So this is an entirely quite complex thing. So what we will do is we will not talk about it. And we will see the brain function as we go during the presentation. So we will uncover some decision-making phenomenon, and we will see which area of the brain is associated with that decision function. And then we will have a more bottom-up approach. So we see which brain is related to what decision-making phenomenon or decision-making problem. And if you really want to know the brain, then there are, I think, many online courses that you can take, in particular on Coursera. That is for free, and I cannot register course there. I think you are just feel free to go there to learn to watch. And the decision part, also in a nutshell. And then there is also a long history of decision making. So what is that decision making? Well, we know intuitively there are options, then we pick one of them, then this is decision making. And so this is quite relevant to our everyday life, right? Here are a few more examples. If you want to unpack the decision-making problem that I described to you, I have options and I have to choose one from them. But the very first thing is you have to see the options. You need vision. So here in this case, do I see red or green color of the traffic lights? So this is your signal. So this is your input. Whenever you see there's a traffic light, and then your visual system will detect the color, and then you will decide whether you want to go across the road, the street or not. So here the decision making is whether you move or you stay there. And some other example can be, shall I bring the umbrella tomorrow? This week is particularly so the case that it's raining a lot. Do I bring my umbrella or not? And what do you do? So you check your mobile app, you listen to the news, you pay attention to the forecast. And then you will use this information to decide if or not you want to bring an umbrella tomorrow, if you want to go to a grocery shopping. Otherwise, stay at home. And some other example can be, am I going to smoke the next cigarette? If you do, even though that's unhealthy. So if I make the same decision sufficiently many times, it may kill me, right? So this is the situation. So if you smoke too much, then it will have really a harm outcome to your health. So that's the decision if you want to take it. A next one or not. Then a further example can be the committee has to make a decision, but the members have different options. And now this happens every day in the government. So the government decides, well, maybe we want to extend the lockdown, maybe not. And then different parties, they have their own different opinions, you never know. So how do they overcome these disagreements? And then they release the news the next Monday, if they want to extend or if they want to finish the lockdown or not. So this is really relevant to our everyday life. There are some simple elements or like ingredients, a recipe, as if we are cooking, right? There are some factors, small parts that we want to pay attention to when making decisions. As I said earlier, how to make decisions. First of all, you need information input. You have to see it. And then there are multiple choice options available. So here there are multiple choice options that you can choose from. It can be brownie, it can be the smoothie, it can be green lights or red light that you see, and then you decide whether you want to go across the street or not. Like in this restaurant example that we had in the beginning, if I want to go to restaurant, I go to Google, and then Google will give me multiple suggestions, and I will use all of the pieces of information to make my decision. And remember how that is called. If you forget, these are called decision option choice attributes. So the name of the restaurant can be attribute if that sounds Italian, authentic or not. So this rating is an attribute. And then the cost, the money is an attribute, and how long it will open is also an attribute. If you want late food, then you will only consider the restaurants where the kitchen is still open, right? So all of these are decision attributes. And another thing that we could consider when studying decision making is people usually choose in a non-random way. Non-random means if, let's say, in the example of crossing the street or not, it's not that I go 50% of the time to the other side of the street and 50 another of the time I stay there, I don't go. It's not the case. There's a system. So if you see a green light, you go. Otherwise, if you see a red light, you stop there. So this is quite systematic. This is about decision making. And we want to know the rules, why people do this. So in this particular case, in the traffic case, well, people learn the rules. They have to do this. If they don't do it, they get like tickets from the police. And then there are some other ways. So if you are a most of the time a much more healthy lifestyle person, maybe you just prefer healthy food, and in a systematic way, you just rarely eat unhealthy food. And in another case, if you are a vegetarian or even vegan person, this is your system, right? If this is your system, then you will never eat meat. You will never make the choice to say, well, I want to eat meat today. Never. So here it's also a systematic decision-making, non-random. And as I mentioned in the first lecture, so decision making also depends heavily on your goal, right? Because in another scenario, let's say, even though you are a healthy lifestyle person, but maybe you just finished a marathon, 40 kilometers, you are super tired, exhausted, and hungry. And at the end of the finishing line, and the volunteers, they prepare some food for you. You don't, I guess you won't consider those food healthy, right? But you will just take it because that's your goal you want to achieve. The goal is you just want to bring your energy sugar level back to your regular level after so much effort, physical effort during the running. So behavior is also goal directed. So all of these are important elements or aspects that we want to consider when making decision making. So it's not only that I give you a choice, I give you all the options and you tell me which one you want, which one you like. It's not as simple as that. There are some elements that we can unpack. And this is the importance and also the interesting part of studying decision making. And here, all of the three elements, if you can connect the pieces together, we could imagine that, well, overall decision making or decision theory it's concerned with goal-directed behavior in the presence of options, right? Just a summary of what I said. And now if we want to put everything together, we have both the decision science part and also the neuroscience part, and then we study decision neuroscience. And here I will just briefly skip, and then you can read it on your own. And then decision neuroscience. So we have one goal to understand the brain mechanism underlying decision making, but we have a little bit slightly two perspectives. So here we say in the paper that I showed you, like the science paper 2014. So the goal of the neuroeconomics is an algorithmic description of the human mechanism for choices. And then here combines experimental techniques from neuroscience, psychology, and experimental economics, and also using all of the methods and the models from computational neuroscience and economics. So what are the two perspectives? The two perspectives is simply what do you study? So you could study decision-making problem from a neuroscientific perspective, if you're a neuroscientist, but also you could also study it from a more psychological or economic perspective. So what is the difference? The difference is that if I'm interested in how the prefrontal cortex is evaluating the value of the option when making a decision making, like this is really a neuroscientific question. what experiment I can use, how should I design experiments, right? This goes back to the first lecture, the previous lecture. And then I will ask people to make decisions and then to measure the brain. So this is a little bit neuroscientific driven approach. And then the other perspective is, well, I am interested in decision making in general as a psychologist, for example. And I'm also curious about how the brain works. I'm not sure if I know prefrontal cortex. I just want to know where the brain is related to decision making. I know a lot of decision theory, but I don't know the brain. So this is my approach. So they have the similar goal. If you ask two groups of people, one neuroscientist, the other one psychologist, economists, they do this kind of experiment, maybe they will end up doing quite similar things, but their goal from the beginning is a little bit different. Well, their goal is the same, but the perspective is a little bit different. Maybe for you, it doesn't matter so much, but just to keep in mind that this is interdisciplinary research and many people, they might come from really diverse backgrounds, but actually the goal is the same. Okay. And in the field of decision neuroscience, what are the questions that we usually ask? So here are a few examples. So first one is, what are the computational processes that the brain uses to make economic choices, economic decisions? This is pretty general, right? And what is the neural basis of these processes, and how and where the value and the probability combined in the brain to provide a utility signal? So here, this maybe sounds a little bit unfamiliar to you, but we will talk about that. This is a little bit more specific scientific question that decision neuroscientists can ask, and also they can try to answer. There are some also other aspects when making decisions, your emotion, your motivation. So how do emotion and cognition, they interact to shape decision? And also one more important thing is, we do now in everyday life, not in laboratory, we make decisions most of the time in social context, right? So there's a huge field of social decision making. So how people make decisions under like social context, social situations. Yeah, we'll talk about that later. Yet here, social decision making, and so on and so forth. Okay. Good. Any questions? All right, perfect. Then let's move on to talk about the research methods in decision neuroscience. Wait, I think, let me stop for a sec. I want to do something a little more important. So what I want to do now is to tell you who are studying this decision neuroscience. So we will see a lot of papers from literature during our course, from the presentations, from the students, and you will be quite familiar with who is who, I guess. But I want to give you a general overview. It's kind of a landscape in terms of who are the people that are studying decision neuroscience. So not surprisingly, many of the research centers or research institutes are based in the United States, but there is also a quite large community in Europe, I think, not so much in Asia yet. Okay, so I think there are too many. I won't tell you everything of it. I will give you two examples, and then these two places are really good to follow. So any research from there is great, I think. It's really excellent. So the first one is Caltech in the United States. So here, if you want to do a PhD later in your career, and if you don't mind going abroad, and then I think this is a really excellent place to study. So if you look at the people, I didn't know there are so many, but anyway, okay. So here, the first one, Ralph Adolphs, we will see his name a lot during the lecture. And there is another one here, John Doherty, and he's really also another name of decision-making, decision neuroscience. So Caltech. And then if you think California is too far away, 9 hours time difference even, there's a closer one in Switzerland, in Zurich. So they have a center for neuroeconomics. You see the name, right? They study like neuroeconomics, and all of the people here, they work on decision neuroscience problems, social decision making, maybe not social, risky decision making. And the interesting part is, so this lab is not based in faculty in psychology. It is based in faculty or Institute of Economics in Zurich. And if you know a little bit more of the background, so Zurich is like quite business bank oriented a little bit. So their department of economics is quite strong, and so that's also why they like established the center, I think, more than 10 years ago. And let's say also that if you just feel overwhelmed about all these centers, is there a like a central place where you can look for people, look for labs, to look for papers? So yes, there is a website. So there is because this field is getting quite interesting and exciting and engaging. Then quite a few years ago, they decided, so many of the people in the field, they decided to establish a society for this purpose. I can now send the link here to the chat. So they have, if you want to see the most recent advancements of the field, they have annual conferences. It's quite expensive to go, and if you are doing bachelor and master, it doesn't make sense to go to such a conference like that. Only if you are doing a PhD maybe, and it makes sense to go. But whatever, their conference like menu, the agenda, this is very interesting to see. So there are the titles of the research, and you can see what are the most recent ones. That's pretty interesting. Most of the time, this conference is based in North America, but here only once, I think, 2016, it was in Berlin. All right, just a few kind of information if you are interested in what or where you can get more information on top of the lecture that we have. So we have not so much time for everything. We have only like 10, 13-ish lectures, but there are like really a huge world of it. What can be more interesting, I think. Yeah, what can be more interesting is actually, so that the Caltech Institute, because of COVID, kind of, and they initiated the online lecture series, and everything is about decision neuroscience. And then also the even nicer thing is they post everything online. So from, I think from here, this one, can you see this? This one? From this one. So this is decision neuroscience lecture, and then this person, the speaker, is the director of the Zurich Center of Neuroeconomics. And then there are other people. I think just everyone studies decision making, and then those are the most recent research. Some of them are not even yet published, and it's really interesting to see. What I will tell you, outdated. Honestly, I want to be frank. So frankly speaking, just a little bit old. Kind of everyone knows it. It's good to know for you as a bachelor student to see how the field has developed. But if you really want to see the advancement, then go to these video lectures. Those are really great. I would say one of the best in the world. All right, so now let's go back to decision making, decision neuroscience and research methods. Say, well, you're interested in those lectures on YouTube and you want to understand many of them, but you have never heard or never used of some of the research methods. So then don't worry, I will get you covered. Research methods, the very first one is actually not so neuroscience, it's actual behavior. So if we study decision making, I give you choices, I give you options, you give me your choice. What is that? That is behavior, right? Behavior is the most important readout of my experiment. We do experiment, we want to measure how people behave in one situation, how people behave in another situation, how people learn, how people do under pressure, how people behave under social context, right? Their behavior is just behavior. So behavior actually matters a lot. And then there is an entire issue 2 years ago in the Journal of Neuron dedicated to behavior. So here, this is the editor of Neuron. And then they have a central claim of that issue is behavior matters. Behavior is the most important readout. And only when you have a meaningful behavior, then you could measure the corresponding neural activity. So in the opposite scenario, I give you like 5 situations and all of your behavior is random, 50, 50, 50, 50. How much meaningful information I could extract from it? Nothing, right? So only when the behavior is meaningful, then the brain response is interesting to see. Otherwise, it's kind of difficult. So behavior is the only most important thing that I want to address. And it is indeed a powerful signal than to the rest of the brain. So behavior, okay, we understand the importance of behavior. What is actual behavior? Yeah, so what are the behavior that we can measure? In laboratory, what we can usually do is we record button press. So many of you told me that you have been a participant of an experiment. So then you just went to the basement maybe, and also somewhere. And then there is a computer program, there's some screen information showing up. You either do a memory task, learning task, like language task. You will make a button press eventually. So here, those are behavior of you. It can be a binary choice. It can be smoothie versus brownie, A, B, 1, 2 binary choice. It can also be on a scale. So after decision, I also ask you, how happy are you feeling at the moment? 1 to 9, you give me your score. This is rating. This is also behavior. And some other people, they are also interested in confidence rating. So if they are making a decision under uncertainty, they make a choice, maybe they're not sure about the choice they made. And then they could also provide their confidence rating. Am I confident about my choice? Am I not confident about my choice? And what else can be interesting? So reaction time, right? If I give you brownie versus smoothie, some people maybe just take it immediately, say, okay, brownie. And some people, maybe they have a difficult time in deciding which one they want to choose. They are a little bit hesitant. And then in this case, the reaction time is longer. So reaction time is also a behavior window to look at. And Here, the three down below, they are not directly neuroscience approaches, but also not common. Well, it's becoming more and more common, but not the standard behavior measurement. So we could measure using some other modalities. Like this can be categorized into an umbrella term called process tracking. So what is the process? So the assumption is like this. If I tell you to make a decision between chocolate brownie and smoothie, you press the button, you tell me A and B, and I could also measure your reaction time, how fast or how slow you are making it. But you might imagine there is some in-between processes that is unknown to the researcher. So what can that be? So think about it. If I have a screen, I give you a screen. So imagine there's a screen in front of you, black background, and then two pictures, one on the left side of the screen and the other one on the right side of the screen. So one of the pictures is smoothie, the other one is brownie, okay? So think about it, envision that. And there are two buttons on the keyboard, just left and right on the keyboard, for example. So before you make the choice, right, what do you see here? There's some visual information. You see, okay, there is a left brownie, there is a right smoothie. What can be interesting is your eye movement. So before you even make your choice, if you don't make your choice, I can never know your choice. I can never know your reaction time. But if I have other measurements like eye tracking, I could measure how long have you stared at the brownie, how long have you stared at the smoothie, or maybe you also jump in between of the two options, right? Your eye movement is a really important information. So here, eye tracking is one of the most frequently used methods in studying decision making. So people want to know how much they look at it. So if, let's say, we could use the other example of the restaurant example. I ask Google, I want to go to Italian restaurant. There are the names of the restaurant, the ratings of the restaurant out of 5, then the dollar sign, the euro sign, how expensive that is, and also the closing hour of the kitchen. So they are properties. I give you 2 restaurants, both of them, they have the 4 properties. Your eye movement will be a lot in this case. Maybe you will compare side by side. You will compare the name of the restaurant side by side, A and B, and then the rating of the 2 restaurants, and then the cost, the expense of the 2 restaurants, and also the some people do this like side by side, but maybe some people they do it another way. So they finish all of the 4 properties of one restaurant and then they go to the 4 properties of the other restaurant. But maybe some people they just do it not in a systematic way, just jump, right? So all of those eye tracking information are very important, and if you do not measure that, you will just lose it. You will only have the decision. Okay, restaurant A, reaction time 2.5 seconds. That's it. It's interesting, but if you have other modalities, the data is much richer. The data can tell us more about it. Similarly, there's something called mouse tracking. So mouse tracking in this case is there is also the screen, and you're not using button press to make the decision. You're using your mouse, you're using your cursor to make the decision from the bottom to the choice. So if you really want the broccoli, you just go straight. But maybe some people, they want to first go to the brownie, but maybe they change their mind, whatever reason. And then you see here the trajectory. So this is the mouse tracking. And the effort tracking the same. Some researchers want to measure the motivation, the physical motivation of making a decision. So if I give you option, if you really want, and then I give you another accompanying option saying that if I give you apple, for example, if you want the apple, you press as hard as you can until you reach the bar and then hold it there for 3 seconds. So imagine if you really want the apple, you are willing to do this, right? And if I am giving you something else, I'm giving you whatever, something less interesting, you don't want to put the effort, and then you don't want it. So here, effort tracking is also an important measurement. And moving next from behavior measurement, what else we can have? We can have physiological measurement. So what are the physiology regarding your body, and just not the brain yet, but some other physiological measurements. So we have the heart rate variability. So maybe if you're under pressure during a decision making, your heart rate will be high. And if you're under threat, so like in the case of electric shock. And then maybe one step back. So I give you two options, like the classic conditioning. The two options, they have no meaning to you, but one of the option is associated with electronic shock, and the other one is a safe option. So one is like painful option or threat option that you could imagine. The other one is a safe option. But before that, you don't know. And then through learning, you could work out that one is more threatful, the other one is much safer. And now the question is, I give you one of the option, maybe the threatful option. When you see it, before you make any choice or anything, if I could measure your heart rate variability, maybe people have a higher heart rate variability when they are seeing the threatful option, and not the case when they are seeing a safe option, right? So psychological or psychophysiological measurement is also quite important. Relatedly, we could also measure the facial EMG, so something like this, to measure the muscles and also the movement, especially when people are perceiving emotions. Some people, when they see a picture, they want to mimic the emotion. If they see a smell, they want to mimic smell. If they see a like sadness face, they want to mimic it a little bit. So then there are research methods to attach a few electrodes on the face to measure the facial EMG. So we have the equipment here in the faculty. And some other things can be like skin conductance response. So the earlier example, one threatful option, the other one is the safe option. People could also measure the skin conductance response. Usually it's measured by attaching kind of a small electrode on the finger. And cortisol level can also be measured, like if you study stress. And cortisol level is very important, and this can be done by collecting the saliva sample, and so on and so forth. Now we could move to a little bit more about the neuroimaging methods, a little bit more about the brain. So maybe I believe that most of you have seen this picture or seen this figure during your earlier study. So what do we see here? And usually, or most of the time, if you see a figure, you have no clue what it is. What do you do in the first place? You will find out what the axis mean. So here we have a simple plot in terms of the layout. We have the axis, the time. We have the y-axis, the size. So one is the temporal dimension. So here the temporal dimension, and here this is a spatial dimension. So the temporal dimension goes from the millisecond, the second, minute, hour, day, month, a week, month, year, maybe. And here the size. Are we entering the synapse layer, or the resolution, or the dendrite, the neuron layer, the column, like the map or regions, or the entire brain? So how precise we can go to. So these questions this plot answers the question about how precise each neuroscientific method is regarding their spatial resolution, how small they can go to, and temporal resolution, how fast they can measure, right? There are multiple, right? And we won't talk about all of them. We will talk about a few. So we will talk about this functional MRI, and these are the tools that we will see a lot during our lecture, during our student presentations. And then we will also see here the MEG and EEG. These are the tools that I will talk a little bit more about. So from your earlier study, I guess you have heard that the MEG and the EEG, they have a high temporal resolution. So they could really measure milliseconds, but they have a relatively lower spatial resolution. In particular, that they cannot really measure brain activity in the midbrain. So because like if you know MEG and EEG, we'll see that there is a cap and it measures everything that's going on the surface of the brain, right? So the cortex. If you want to measure something deeper inside, then EEG and MEG doesn't go this way, it doesn't work this way. So this, in terms of this spatial resolution, that's a little bit limited. And the functional MRI or MRI, in contrast, it has a higher spatial resolution. It goes here. It can be as precise as millimeter, 1 millimeter by 1 millimeter, like small cube. I will show you. And then they go everywhere in the brain. But the temporal resolution is a little bit not so ideal. Okay, I will tell you both of them. But before that, I tell you a little bit more about what are the most important neuroscientific methods. That is not so usually you can't do it in human. You can only do it in animals. So this is single unit recording. So if you want both the spatial resolution and the temporal resolution, you just want to continuously record it. So what do? You just insert an electrode into the brain. You have to do surgery, I guess. And then you insert the brain here, insert the electrode here, and then you will see the most precise and the cleanest brain signal. Single neuron recording, animals. So here in this particular case, I won't go into too much of the details. There's a learning task, Pavlovian learning. There's a cue, and then the mouse is trying to associate the cue with the outcome. So in the beginning, the cue is not associated with anything. And then during training, if they see the cue, they will receive a reward. So usually food, cheese, and juice is a quite delightful reward for animal, for mouse. So if they see a cue, then the cue is associated with the food. They will learn this association. And then in the next phase, they will only see the cue again, but without being delivered the food reward or the juice. And so how would you imagine this case if you are interested in these brain processes? And what we can see here is at the beginning, when the cue is not yet associated with the food reward. So here on the x-axis, this is the time. This is the duration of the experiment. So maybe here when they see the cue and then there's some time in between, there's the delay. And then here, this is the moment when they receive the reward, and then also a few seconds afterwards, okay? X-axis is the time. The y-axis is the neuron recording, the spikes of the neuron. So each row corresponds to one neuron. And the dots here means there's a spike, there's a signal. So when it is white, then that means the neuron is not firing. And when it is black, then that means there is a neuron firing, the neuron fires. So then what people can do is to aggregate the neural activities. So here at the third, here, this is the third moment of after seeing the cue. So some neurons are activated. Yeah, most of the time, not really. And most importantly, after the food reward is delivered. the neuron is really firing because there are a certain area in the brain, in this case, the dopamine neurons in the midbrain. So the dopamine neurons in the midbrain, they are like responding to the reward immediately after the reward is delivered. Great. And then later, what else they can measure in the second phase, I told you, is when the cue is associated with the reward. The important part is if the mouse is seeing the cue, they will already anticipate a positive reward. So that means the cue itself is rewarding. So the interesting part here is when they see the cue, results even before the reward is delivered, the dopamine neuron is already firing because I could anticipate some reward in the near future. And whenever I see the cue, I'm already feeling rewarded. This is a lame explanation, but there are some, but more or less the idea is like this. And more interestingly, so here, if I see the cue at the very late of the experiment, when the association is established, the researcher, they remove the reward of the cue. So they give the animal only the cue, but without giving them the reward. This is the interesting part. So when the animal is seeing the cue, the dopamine neuron is still firing because they might anticipating there is something good happening, there will be the juice delivered. But at the moment, approximate the time previously they were getting the food reward, but in fact, they didn't get it. So here, the firing rate is even less than the baseline, right? So here, there's something negative happening. Negative means the actual outcome is worse than my expectation. So there's something called a prediction error. We will see this a lot. So here, this is a negative error. And then the dopamine neuron is also associated with the negative prediction error by reducing the firing rate. Okay. This is a really longer story than I expected, but I think this is quite important because this paper is a science paper from quite a while ago, 25 years even, more or less. This paper is really the influential paper in decision neuroscience because it has the first evidence of showing the dopamine neuron, midbrain dopamine neuron is associated reward prediction error in this case, I showed you. And it also corresponds to learning, right? Where's the learning? Well, the learning is here, the second part, and so the third part. When they establish the association and when the association is validated. So this is like learning phase. This paper, I think it is cited, I don't know yet, I didn't check the stats, but I think 6,000 or even more, 7,000 times nowadays. It's really influential. So you might wonder, single neuron recording, well, it's great. This has been done nearly 25 years ago. What are the advancements? There are indeed actually advancements. It happened yesterday. So there is a new paper in Science as well. So what they have done is they could have a longitudinal recording of the animal, free moving animal. The animal can be in an arena, they can do whatever they want. And then there's some device attached to the brain. And then they could measure the brain signal in a longer time duration, I think 3 months. And you might imagine, well, what is the challenge here? I just measure it and then it takes 3 months. Okay, maybe time taking 3 months is not so ideal, but I can still do it. What is interesting? Well, the interesting part is here. So if you measure the brain activity, usually you won't let the animal wear the entire device for the rest of the day. And then you just have to take it. And then the animal, they also have to sleep. They have their life, right? So now the question is, if I measure it, I take it off, I measure it again, how can you make sure I am measuring the same area, right? Maybe a 1 millimeter shift about thousands of neurons. So this is challenging. And I think now those people from England, they are really solving this problem pretty well. And this is the advancement. So next question. Single neural recording is so useful. Can we do it in a human? No, yes, no, kind of. So healthy human, we can't. But patients, yes. So usually there are some patients you have to do a brain surgery nonetheless. You have to open the brain because you want to do a surgery to treat the patients. So in this kind of in situations, most of the time researchers are collaborating with the neurosurgeries. And then those patients, when their brain is being operated, they can also do some tasks. And then the neural recording could also apply to human in this case, only in this case. Otherwise, well, there's ethical issues. Right? All right. Oh, well, here, this is already, this was 2 years ago, I think. Now I think it's even more, 9,000 or maybe 10,000. Great. And this can also apply to monkeys, well, any kind of animals. Most of the cases is mice and rats and monkey. Mice and rats they are different actually. So one is smaller, one is bigger, and also monkey. And most of the time, macaque monkeys is the common subject of decision making. So in this particular case, you can see here, the brain here, this area is being recorded, and then the animal is making a perceptual decision making, just colors, and then they will also see the screen, and then they make a decision. I won't go into the detail, but in any case, single neuron recording is used a lot in animal research, animal decision making. Yes, one small thing. So when we talk about decision neuroscience, it's not only about human. So even in the case of neuroeconomics, we think about that, well, humans make economic choices. Well, this is also true in monkeys because they are smart enough and then they know the utility. They could know, well, if something can benefit themselves, if something is not, right? Decision neuroscience, I would say roughly 70%, 60 to 70% of the time, human researchers, maybe less, let's say 60% of the time, human research, and then the rest, animal research, mostly in monkeys. And this is a little bit more perspective that I'm telling you, because I think if you're like doing your bachelor, knowing these kind of things maybe is interesting, but maybe not. What is more important for you to know is to see, well, how science works, right? You have a bigger idea of what the field is looking into, what are the interesting questions, and what are the field that you want to go to in the future. Either you want to stay in academia or you want to go somewhere else, it doesn't matter. There are some like transferable skills that you can carry on. So here, one perspective that I can share with you is that, well, you would imagine that people do research a lot, decision-making questions, decision neuroscience questions, 20, 30 years already. Are there new questions? Well, the question is always yes, and the brain is so complex and it's so fascinating to study it. And then how can you come up with new experiments to study the brain more sophisticatedly? One kind of approach is to always, my personal opinion, is always to go to the animal literature, because there are many things you can do in animals, but you can't do in humans. So animals, the research, I think, is most of the time a little bit advanced relative to human research. Not always, but a lot of the times, because they can do single neural recording, right? This is something you can't do in human easily. So if you look at the animal literature and then find something interesting in animal, but no one has done that in human, yeah, then you have a field. Then you know what to do next. Kind of a shortcut, not really, but kind of. Yeah, see what I mean? Okay, so 5 minutes left. I won't finish everything. Let me just finish this. Some advantage and kind of disadvantage of single neuron recording. This is the only direct measurement of neuronal activity. Really, you directly measure the neural activity of the brain in animal or in patients, human. It's invasive, obviously. You need an operation to attach electrodes onto the brain. And it is a little bit limited to the spatial resolution. What that means, that is, so you have a kind of, usually this is called a recording array. So it can be 5 centimeter by 5 centimeter. This is already a large one. Sometimes can be smaller. This is not large. If you attach it here, then you can only measure prefrontal cortex. If you measure it here, you can only measure the parietal cortex. If you measure in the back of the brain, you can only measure your visual area. So what this means? This means you have to be precise. You need, as a researcher, you need to know where you are going to measure. So like in the 1979 paper, the prediction error paper, I told you they are measuring the midbrain dopamine neuron, but why they decided to measure midbrain dopamine neuron rather than some other areas, right? This is important. So using single neuron recording, you do need to have some knowledge of the brain. You need to be precise of your hypothesis. You hypothesize here midbrain dopamine neuron is related to prediction error, then okay, go there to measure. But if you have another assumption, then maybe you either don't get it or maybe you get it a little bit differently. Okay, this is really brain area driven, a little bit limitation. But if you get it, you get the most precise and the cleanest signal in the world. And there are some, I think this is review paper. Yeah, whatever, you can forget about that. Yeah, this is interesting read, I think, but if you don't want, you can skip it. So the remaining 3 minutes, I can briefly talk to you about brain lesion. So brain lesion is easier to explain. So brain lesion means there is a brain damage in the brain, in the patients in particular. So for whatever reason, there is a brain damage. So their area of the brain is different relative to a healthy participant, healthy people. So here in this case, these two like plane, they are taking a slice of the brain. So here in this case, we could see something in the middle here in this slice. It's a thin slice. And then here, this is the view of the brain. On the left-hand side, this is the healthy participants. And here, this is the patients. What you can see here quite clearly here, this part relative to here, there is the brain damage. So this is the brain lesion. And then this particular area is called amygdala. So if you have maybe some knowledge about the brain, then I guess you have heard about that amygdala is very important in processing emotion and also in terms of recognizing emotion. And if I know that amygdala is associated with emotion, then if I'm lucky enough to have amygdala lesion patients, I can study a lot with the patients. So I can study if the patient can recognize emotion, if the patient can do some mathematical tasks equally or not. So the idea is that that only affects emotion or cognition in general, right? I can really do a lot. So in this particular case, then they ask the participants, one healthy and one lesion patients, to recognize the emotion. I show the patients the picture, happy. So here on the x-axis, happy, surprised, afraid, and angry. I show you the face, the facial expression. You tell me what that expression is, and like just making your choices down below. Here the circle is the healthy control, and then here this triangle, and this is the patients. So what you can see here in terms of the positive emotion for happiness, no difference between healthy and patients. And then here for sad, this is also no. But what is interesting is really here, kind of surprised and afraid. And here there's a huge difference between the healthy and the patients. And then this is some first evidence of amygdala is responding to some of them, some specific emotion. And here, if you still remember the name, Rolf Adolphs, so this is the person that I showed you on the Caltech website. So he was the person who is doing this like nearly 30 years ago. And quite also importantly, remember eye tracking. I can measure how aware do people look at when they are seeing a facial expression. So the idea is that healthy people, they tend to use like the eyes to identify emotion. And then the patients, they maybe avoid looking at the eyes. Instead, they look at the nose. And then this is less informative when you are trying to detect, to identify, to recognize an emotion. So here, this is a multi-combined research. You measure behavior, you use eye tracking, and you can also measure some others. Okay. So brain lesion usually very rare. So that's why I said if I'm lucky enough, I can do a lot. But what if I never know a brain lesion patient? So then I can never know. I make that up in this way, right? And the brain lesion is causal. So if I know the brain is there, it works. And I also know that if when the brain area is not there, then something is not happening, then this is a causal causal inference. Then this area is really the cause of the consequence, the causal inference. Brain lesion is for that. And the consequence, a long term means, well, if there's a brain damaged person, maybe this is carrying over throughout the end of the life. And maybe for light, not so severe brain lesions, the lesion or the damage can be compensated by brain plasticity, but maybe not. Yeah, this is the causal idea that I'm emphasizing. So here, this is also important, a research method to bring some insights. And again, Adolf, so he wrote a paper 2006. So he was reviewing some of the recent findings of brain lesion studies in 21st century. So feel free to take a look. All right, I will stop here and then we will continue next week about the remaining neuroscientific research methods. And any questions so far here?